So let's talk about the exams. As I mentioned, this tends to be one of the more challenging exams, and this also tends to be one of those tests, again, where there's a huge range of scores, so the averages aren't necessarily indicative of how the class did. While the lab exam score and lecture exam scores are relatively low, almost half the class got A's and B's, which tells you how the other half of the class did, if these are what our averages that we're looking at are. There are three pieces of good news. The first is that your grades in this class are not based solely on your exams. As I said, all the other work that you're doing goes a big way towards helping it. And so our average in the class is still over a C, which is nice, but it definitely needs to come up. Second piece of good news is that I think the two hardest exams are now out of the way. The first one, because you don't know what to expect. The second one, because like I said, it's the least intuitive information with all that histology that we're dealing with. There will be histology on other exams, but none of the other exams will have as much histology as we saw on this one, because it was all tissues. Even the skin, our first organ system, was just all tissues. So hopefully we'll get a little bit more intuitive that way, and so make it a little bit easier. And then the third thing is that, again, there is still time. You now have had 2 lab exams and 2 lecture exams. You know exactly how I'm going to ask the questions, what I'm looking for information-wise. If you're not getting the grades that you want, I strongly, strongly encourage you to come to my office hours today or sometime this week so we can discuss your exams. Many of you need to look over them anyway for your science success centers. I've gotten emails from you guys. So let's make sure you know where you need to improve and what you can do differently to get the grades that you want, because there is still some time and lots of wiggle room on these exams. Some of the things that are going to help us to be successful is moving forward in this section. And in particular, again, you've got that 30-point skeletal review that is going to be due on the 13th. It is graded for correctness. So that is a big, huge chunk of points that you want to make sure you're successful on. And to help you to be successful on that, we're going to be going over the bones and bone features starting today. Today, we'll start with the axial skeleton. Like I said, my goal for today is to get through the skull, which means Wednesday we will get through the rest of the axial skeleton and then also assign you your appendicular bones, because next week we'll be doing the exact same thing. You'll be doing bones and bone features on Mondays and Wednesdays for that. Monday will be the upper appendage, and Wednesday will be the lower appendage. So you'll know exactly where you are in the presentations for those. All leading up to the 18th. So you've got about 2 weeks. Actually, you have exactly 2 weeks till the next exam. So lots of time to study and prepare and to get ready. All right. Questions on any of that?
I have a question.
Yes.
I know that you said that with the PowerPoint presentations, because we're going to be speaking, that it's not going to be recorded.
Yes.
Is there a way that we could like upload our PowerPoints so we can, like, if somebody wants to study, like, I don't know, like the typical ribs or something like that, they can go through the PowerPoint itself without actually having to go through lecture, like in a file?
Really, that part of it shouldn't be necessary, because if you have any type of atlas, like you should have, I know this isn't the particular atlas that you have for your class, but any of the atlases that you should have, and you should have an atlas, is going to have the bones and have the bone features on it. Your textbook has the bones and has the bone features on it. Your lab manual has some of that material, the PAL CDs. So all of those things have those resources so that when you want to study it on your own, you have the opportunity to do that. But I think sometimes with us going over it together, there are some tricks, there are some tools that can be used to help you to be able to better identify those things. And that's kind of the point of this, is to expose you to these things, kind of try to give you some tricks to help you to be successful, so that as you're working on it on your own, that you can do that, because that's the whole point. After having one group present one bone for 5 minutes, I don't expect you to have mastered the bone after that time. we are going to need to spend a lot of time and a lot of work over the next couple of weeks mastering this material. But getting that exposure at the beginning is something that can be definitely very beneficial. So that's kind of the point of this. We're exposing you to it, introducing you to it, so that as you're studying this on your own, you have a better understanding of what you're attacking the material with.
Got it.
Yeah. Go ahead, Ava.
Do you recommend starting the 30-point skeletal review like the weekend, I guess, before it's due, or after we complete that Monday of class?
It really comes down to more your level of comfort. Like I said, the whole point of this skeletal review is to take the bone and bone features and put them together to understand how they relate to each other. So like I said, the simplest example is on the humerus. There is a big bone feature called the trochlea. On the ulna, there is a big bone feature called the trochlear notch, right? And so not surprisingly, the trochlea of the humerus sits into the trochlear notch of the ulna, right? Now they happen to be named in such a way where it kind of shows that they're going to be related to each other, but that's the point. You need to know how these bones relate to each other. If you don't know what the trochlea of the humerus is, if you don't know what the trochlear notch of the ulna is, then you're not going to be able to understand that relationship to each other. So you need to start at least familiarizing yourself with the bones and bone features before you can start putting them together. So that's why I've got to do on the very last day of class. But when you start gaining that level of comfort, and that's the thing, you don't have to do it all at once. Once you do the arm bones, if you want to then look at the bones, or once we do a particular bone, you can go through it and do it. So you can do it bit by bit as you get familiarity with it, or save it all till the end. But when you start is completely up to you. If you wanted to start now, I think it's available now. And so now you actually can do that. But I find that most students beat their head against it if they don't understand the information. If you don't understand the pieces, you can't put them together into the big picture. And so while you could brute force your way into doing that, you're not really learning anything by doing that. And so it's not conducive. But again, it's you're grown-ups, you're adults. This is college. If you wanted to do that, you can do that. So however it is. Okay. All right. Any other questions? All right, excellent. So last thing I want to mention, as I talked about in the last class, now finally we're going to start seeing some division in the class. So in lecture today, we are going to focus on our bone physiology, and then in lab, we're going to be doing our bones and bone features. So we're really going to start seeing some dichotomy in this information. So let's go ahead and dive into that bone physiology. As I mentioned, when we look at a skull, when we look at a bone holding it in our hands, it looks like this just dead hard piece of tissue. But as we also talked about, bone is incredibly dynamic. And being incredibly dynamic, there's a whole lot of processes involved in this. Obviously, the first thing we need to do, starting in utero, when we're that embryo, when we're that developing fetus, we need to make bones. Once we make bones, we then are born, and then none of us are the same size now that we were when we were born, so we need to grow bones. And here's two other things to remember about this. As it turns out, there are going to be two ways to make a bone. So remind me again, how many possible essay questions are we going to get out of that?
Three.
Three essay questions. What are they?
Describe A, describe B, and compare A and B.
Exactly. Describe the first process, describe the second process, and compare the two processes. Absolutely. All right. There are two ways to grow bones. Any idea what the two ways to grow a bone are?
Is one interstitial?
Okay, you are correct. That is a growth process, but that isn't really what I was talking about. That's why I didn't do it for the normal bone growth, because I don't want to worry about phrases so much. You have a tiny little baby bone and you want it to be an adult bone. What do you have to do to it? Yeah, exactly. We want it to grow in width, but so not get it fatter, but what do we also want to do? Are we all just really
Grow in
short people
length?
with huge fat bones? No, yeah, we want to grow it in length. Exactly. So we want to grow it in length. We want to grow it in width. And so, of course, two ways to do that. So, of course, 3 essay questions. Excellent. Now, as we also talked about in the last class, I think most of us have reached or are about to reach that point where our growth plates close and you're not getting taller anymore. If you're not getting taller anymore, does that mean that your bones can just check out and they don't have to worry about doing anything for the rest of your life? No, of course not. There's a tremendous amount of dynamic growth processes that go on with our bones. And so we're going to talk about ways to maintain the bones. And as it turns out, there are going to be 3 ways to maintain the bones for that. And then, of course, also, as we talked about, if we damage the bone, we then need to be able to repair that tissue as well. And from what you know, based on the last question, there are 2 growth processes. What were the 2 growth processes we learned about in the last section? There we go, fibrosis and regeneration. Excellent. And which of those 2 processes for healing do you think take place in bones?
Regeneration.
Regeneration, right. When we damage the bone, we replace it with bone connective tissues. So it is indeed a regeneration process. Excellent. However, the other thing we need to talk about in relationship to bones, as we mentioned, is that vital importance of calcium, right? Obviously, we want to put as much calcium into the bones as possible to make them big and strong. In fact, as we talked about, 99% of the body's calcium is stored in our bones. However, if your interstitial fluids, if your blood starts lacking for calcium, is your body just going to say, that's okay, calcium's not that important, we can have low levels of calcium? Or is it going to actually go to the bones and damage the bones essentially by taking that calcium out of it to make sure that blood calcium levels stay high?
Yes, it takes it from the bone.
Yeah, absolutely. If it comes down to a choice between having dense bones and having the right amount of calcium in the body, calcium is going to win every time. So we need to talk about that role that calcium homeostasis plays in the affecting the bones as well. All right, excellent. So for the next 2 weeks, this is our lecture game plan. These are the things that we're going to be talking about. And so let's start first by talking about how we make bones. Now, if you remember, as I mentioned, the way we make bones involves 2 processes. And when we're looking at that developing embryo, the developing embryo at first doesn't have bones. Instead, it has 2 different types of connective tissue. It has a fibrous connective tissue, and it has hyaline cartilage. So, 2 ways to make bones, 2 types of connective tissues. Do you think that that's just coincidence? Do you think it's coincidence that we have 2 different types of connective tissues in the embryonic
One's for
skeleton?
our cancellous bones and one's for compact bones.
Not a bad guess, but no, but it's good. You are the right idea, though. It is going to be used for making different types of bones. The way we make a bone in a fibrous connective tissue is going to be different from the way we make a bone of hyaline cartilage. And we are going to make different types of bone. But remember, now we're talking about making organs. So we're not talking about making, the spongy or the compact bone. Those are the connective tissues. We are making organs here. Remember, at this point, 2 processes for making bones the organ. Excellent. So starting early, about 6 or 7 weeks of development, we start to convert that fibrous connective tissue, convert that hyaline cartilage into bone. And there are 2 ways that we do that. For the hyaline cartilage, we use a process called endochondral ossification. Ossification is the process of making a bone. Endo means what?
Inside or in?
Inside, and chondrial refers to what?
Cartilage.
Cartilage. There you go. So within the cartilage, we make bone. And the second process is intramembranous. What does intra mean? If you do intramural sports, are you going to other campuses to participate in that?
No.
No, intra means within the walls of. So intra means also kind of within. Membranous, within the membrane. So we are making bone within a fibrous connective tissue membrane. So there you go. 2 processes for making bones. All right. We are going to talk about endochondral ossification first, but before we can do that, we do need to talk a little bit about how we're going to make our hyaline cartilage. Basically, what we do is we have a chunk of hyaline cartilage in the shape of the bone that we want, and we're going to use this to make our bone. This is how we make all of the bones of our body, and all of our long bones. Now that's good because we know the long bones have a very fancy anatomy, and so we can pay attention to that. And like I said, it's going to start with, put that in the wrong place. Up there. So that way I can do this. To remind you that before we can talk about endochondral ossification, we need to talk about how we make this chunk of cartilage that is going to be our bone starting point. As we know, cartilage, like all connective tissues, come from a pluripotent stem cell known as a mesenchymal cell. It can divide to become any of the cells that we want, and it divides to become chondroblasts. Chondroblasts make a chunk of cartilage, and as we know, that chunk of cartilage is going to have a dense, irregular connective tissue around its outer surface. And what would we call that dense, irregular connective tissue around the outer surface of a cartilage?
Perichondrium.
Perichondrium. And as we know, that is going to house those mesenchymal cells. So we're going to have those mesenchymal cells housed in here. All right, so let's switch to the whiteboard. Here we have a chunk of cartilage, a hyaline cartilage. And as we know, that hyaline cartilage is going to have chondrocytes in it. So let's just put one chondrocyte inside of it. And as we just finished mentioning, it is going to have a perichondrium on its outer surface. Excellent. Now I'm going to cheat a little bit and move it away because, as we also just finished talking about, inside this perichondrium is going to be our mesenchymal cells. And inside our inside of our lacuna, we have a chondrocyte. All right, so this is a very simple basic model, but this is our starting point. This is our chunk of hyaline cartilage that is going to become a model for our bone. Now, notice right now it's just a square. It's not very much bone shape. So what we need to do is we need to use 2 growth methods to convert this square of cartilage to form into our bone-shaped model. Now, as we also have learned, life is lazy. right? Whereas, and I think this was some of the things that some of you were confused about when it got to the skin, while regeneration and fibrosis are processes that the skin uses to repair, is skin the only tissue that uses regeneration and fibrosis to repair itself?
No.
No. And it's the same thing here. We are going to look at these growth methods here in the cartilage to gain our basic understanding. But life is lazy. Once it's learned a process that works, it's not going to just save it for one type of tissue or one process. It's going to use it for many, many things. So these growth methods are going to be used for other growth processes as well. Excellent. Now, the first one is what is known as interstitial growth. What does interstitial growth sound like? With a name like interstitial, what does interstitial mean?
Within the walls.
True,
Inside.
or within the tissue, absolutely. So this one occurs inside the tissue. So notice what's going to happen here is, as we mentioned, we have this chondrocyte hanging out in its lacuna. I guess we should label our lacuna as well. Oops. Actually, I'm going to move all these down here. No, not you. Just in there.
GB, no.
All right. So we have a chondrocyte living in a lacuna, just like we saw how a osteocyte lives in a lacuna. But there is one big difference between chondrocytes and osteocytes when they are in their lacunas. And that is that our chondrocyte is actually capable of dividing. Remember, an osteocyte isn't capable of dividing. In fact, the only bone cell that is capable of division is that osteogenic cell. So what's going to happen is we are going to now have two chondrocytes living in the same lacuna. Now, I don't know how many of you have kids, but I have two daughters. And when they were very young, we kept them in the same room. But now as teenagers, are they still enjoying the opportunity to live in the same room with each other?
No.
No, right? They don't want to be in the same room together. And the same thing is true with these two chondrocytes. These chondrocytes want their own rooms. So what they are going to start to do is they are going to start making matrix. And they're going to start surrounding themselves with matrix. And in particular, they're going to start building matrix between themselves. As they start building this matrix between themselves, they are going to be expanding the tissue, basically pushing away from each other, surrounding themselves with more and more matrix, surrounding themselves with matrix in between themselves. And as they're making all of this new matrix, notice what ends up happening is the tissue is expanding, and it's expanding from within. So as a result of this, and I could grab the box and pull it outward to emphasize that, but we are getting this outward push of the tissue, where basically our chondrocytes divide. As they divide, they make new matrix. And as they make new matrix within the tissue, it expands the tissue. Right? The tissue gets bigger. From within. And so in this fashion, our tissue gets bigger. Now, I've done a truly amazing job of showing this, but let's look at the pretty picture from the textbook that shows this exact same process. Notice here, their picture is a little bit nicer than mine. Again, as you can see, we start with a single chondrocyte in a single lacuna. It divides to produce 2 chondrocytes. And as those chondrocytes make new matrix and surround themselves with matrix, notice they are expanding the tissue outward. And what started as a small block of cartilage is now a bigger block of cartilage. So interstitial growth is this expansion of the tissue from within. We divide the chondrocytes, we make new cartilage, we expand the tissue from the inside. Now, this is going to help to make our cartilage bigger, to make it into a bone-shaped chunk of hyaline cartilage. But as I mentioned, growing cartilage to make bones, is this the only reason we're going to use this interstitial growth process? No, and in particular, one of the places where we will see it in this section is in the second step. Remember, as we talked about, once we make bone, we then have to grow the bone in length. Well, this is where we're going to grow the bone in length. This process of interstitial growth grows the bone in length. And remind me again, what is the anatomical structure that allows bones to grow in length? Where do bones grow in length when we looked at the long bone?
The cartilage.
Yeah, that thin piece of cartilage that was in the metaphysis, in the neck of it. And what did we call that?
Epiphyseal plate.
Yeah, the epiphyseal plate. Exactly. So this is exactly what's going to happen in the epiphyseal plate to make the bones longer. Chondrocyte divides, those divided chondrocytes make cartilage push away from each other. And as they push away from each other, the head of the bone moves further away from the shaft of the bone. And so this is the exact same process. This interstitial growth is the exact same process we are going to use to grow the bones in length. All right. Questions about our first growth process? All right, excellent. So let's talk about our second growth process. Our second growth process is appositional. Oops. Appositional growth occurs under the perichondrium. Right? For this process, instead of the chondrocytes dividing, instead here it's the mesenchymal cells that are going to divide. And this is going to make new matrix on the outside of the tissue. So let's grab my perichondrium and move it out of the way even more. Oops, nope, I want to leave that there. I want to expand it out. There we go. Excellent. So as I mentioned, this is a mesenchymal cell. It is going to divide, and when it divides, it is going to produce let's start this here. Going to divide to produce a chondroblast. Let's make that smaller. And of course, what do chondroblasts do?
Make cartilage.
They're going to make cartilage. So this chondroblast is going to start to surround itself with matrix until it completely surrounds itself with matrix, at which point it then matures into a chondrocyte. And does only one chondroblast have to form? not
No.
your question. No, we can form many of them. And as we form many of them, and whoops, they all surround, stop it. They all surround themselves with matrix. Notice in this way, I have now formed a new layer of matrix on the outer surface of the bone, making it a little bit bigger. And again, there's still going to be plenty of mesenchymal cells up here. And as these mesenchymal cells continue to divide and continue to make new cells, then those new cells are going to become new chondroblasts. Whoops. And those new chondroblasts are then going to make layers of matrix on their outer surface. And in this fashion, we are going to continue to get this expansion of tissue, making the tissue longer by adding layers on the outer surface.
When does the cartilage know when to stop dividing?
How does anything know how to stop doing anything? What do you think the primary way is we tell these cells what to do and when to do it?
When there's like an overproduction,
True, but how does it know there's an overproduction?
I'm just relating to like a stop codon, or like there's a process that.
True, but remember at that point, we're talking about molecular processes that are taking place inside of the cell. Remember, we've gone up a level. We're looking at cells. When we have a whole cell, how do we tell that cell what to do? How do we tell cells, if all cells kind of basically start the same way, how do we tell some cells to be toenail cells and some to be cartilage cells and some to be eyeball cells? And
Like
what
signals, our brain?
Well,
Yeah,
they're specific.
signals, but not neural signals. It'd be really hard to send neural signals to every single one of the trillion cells in a body. Instead, Augustina's got it. Hormones, chemical signals. So we are going to have chemical signals in the environment that are going to tell these cells what to do and when to do them. All right, so notice this process expands the tissue from the outside. Right, we are adding new layers on the outside. It turns out, again, life is lazy. We're going to use this process for multiple things. And again, we're going to actually use it again here in our bones. This process is also used for two things. This is what is going to grow our bones in width. The other way we need to grow bones. And this is also how we make compact bone. And of course, someone remind me again, what is the organizing structure of compact bone?
Osteum?
Osteon.
Exactly. So this appositional growth is also how we are going to make our osteons. This is how we make our compact bone. This is how we make our osteons. And again, while I've done a truly amazing job of drawing this, here we see the pretty picture from your textbook showing that exact same thing. Again, here we have that perichondrium on the outer surface with those mesenchymal cells dividing, producing chondroblasts that are surrounding themselves with matrix and adding more new matrix, more new layers of cartilage on the outer surface of the bone, or pardon me, of the cartilage piece, making the cartilage bigger. And again, not only does this allow us to increase the size of our cartilage, but remember, as I also mentioned, we're also going to use this to grow bones in width as well as making our compact bone, making our osteons. All right. Questions on those two growth processes? Notice again, we haven't even talked about making bones yet. These are just growth processes that we're using. We're using them here to make a chunk of cartilage that we can turn into a bone. But remember, we're going to use these growth processes for other things, not just in bones, but as we move on to other systems as well. So questions on these growth processes before we start talking about making a bone. All right, excellent. So now our goal is to make a bone. And notice it starts with a chunk of hyaline cartilage that is in the shape of a bone we want. Now, as we have done in the past, when we have these more complicated processes, I want to go over them together and I want to do it twice. For this first time, I'm just going to do it here on the whiteboard. For this first time, I encourage you to just sit and watch and think and listen. See if it can make sense to you and you can understand the process. Interact with me, watch, think, listen. Then after that, we'll go back to the pretty pictures from your textbook. We'll go back to the pretty words from your textbook and you can write to your heart's content. But for right now, let's just focus on understanding the process. So our goal now is to understand growth process 1, and that is endochondral ossification. And of course, remind me 2 key things about this. We are in endochondral ossification. We're making bones from what?
Within the cartilage?
Yeah, from hyaline cartilage. And what type of bones do we make using this endochondral ossification?
Long bones.
Yeah. In fact, remember, I said most bones of the body, and
So
more
all long
specifically,
bones, right?
all long bones. Since we use this process for all of our long bones, that's a good starting point. We'll look at a long bone. Now notice your textbook picture, right, shows us that beginning point of that dino dog bone, that it shows the entire bone. To give us more room to be able to do this a little bit larger and make sure we can make sense of this, I'm going to rotate this and stand it up on its end. And we're only going to look at one end of it, because everything that happens on one side of the bone is happening on the other side of the bone as well. So what we can do then instead is basically just look at half of the bone here. There we go. And then when we look at this half of the bone, actually, let's look at a little bit more than that. Get the middle of it in there. So we'll look at 60% of the bone. We can talk about this process of endochondral ossification. Now, this is, of course, our hyaline cartilage. So we need to put some of our chondrocytes in here. There's our lacunas. And there's our chondrocytes. Excellent. And we can't forget about our periosteum that's hanging out here on the outer surface. And again, we'll cheat a little bit by pulling it out, because that will give us room to then put some mesenchymal cells in here. So these are mesenchymal cells. And as I mentioned, this right here is our periosteum, perichondrium. All righty. Here's our starting point for endochondral ossification. We have a chunk of cartilage that is in the shape of the bone that we want. It's got a perichondrium, it's got its chondrocytes, and it's been happily growing and making cartilage. as you guys mentioned, and thankfully pointed out for me, the reason it is making cartilage is because of the chemical signals that are present. And those chemical signals are telling these mesenchymal cells to divide and to make chondrocytes. But we're done making cartilage. We want to make bone. So we need a way to change the chemical signal, to bring a new chemical signal into the party to tell these mesenchymal cells to change what they're making. And what would be a good way to change the chemical signals in the environment? All right, let me ask the question this way. What's one of the biggest differences between bone and cartilage? I know you know this. If I wanted to bring a new chemical signal, there you go. If we wanted to bring a new chemical signal into this perichondrium, we would use blood vessels. Exactly. One of the biggest differences between cartilage and bone is cartilage is very, very vascular. I mean, avascular, whereas bone is very, very vascular. And that's exactly what happens. The first step in this process is that a blood vessel, a blood vessel grows in, and I think this is going to be too big. Let's go there. A blood vessel grows into the perichondrium.
The nutrient artery or something?
Not the nutrient artery. We'll talk about the nutrient artery in just a minute. This is just a perichondrial blood vessel that grows into the perichondrium. And as you pointed out, the advantage of growing in this blood vessel is it can bring new chemical signals. And these chemical signals trigger a process. Basically, now with these new chemicals, with this new chemical signals, those mesenchymal cells that were before dividing to produce chondroblasts now produce osteoblasts. And of course, what do osteoblasts make?
They make bone cells.
Exactly. And so that's what happens. This mesenchymal cell is now going to divide and become an osteoblast. And osteoblasts make matrix and surround themselves with matrix. When they surround themselves with that matrix, they become osteocytes living in that matrix. And their job now is to maintain it. And this process will continue. So notice as a result of this, we are going to produce a thin layer of bone on the outer surface of our cartilage. This thin layer of bone is what is known as the bone cuff or the bone collar. All right, so far so good. Now, notice one more thing. This structure here, we called a perichondrium because it was sitting on top of cartilage. Notice it hasn't changed. It's still a dense irregular connective tissue. It's still housing mesenchymal cells, but notice now it's sitting on top of a thin layer of bone. So while the tissue itself hasn't changed, its location has. Is it still sitting on top of cartilage?
No.
No. So notice if it's not sitting on cartilage, it can't be a perichondrium anymore. Now it's sitting on a thin layer of bone. So what happens to it now?
It's a periosteum.
Yep, exactly. So as a result of that, our perichondrium now becomes a periosteum. All right. Questions on this first step? All right. Notice changes are occurring on the outside of our bone. At the same time, changes are occurring inside the bone as well. And in particular, what starts to happen is that at the center of our diaphysis, the chondrocytes grow in size. So suddenly what happens here is these chondrocytes get bigger.
Is that what makes our medullary cavity?
We're getting there. Not yet, but we're getting there. So they grow in size. Now, in and of itself, is there a problem with these cells getting bigger? I mean, it's just a bigger cell. It's inherently not that big of a problem. But obviously, if they get bigger, then their lacunas have to get bigger as well. And notice if their lacunas get bigger, there is less matrix in between these cells. And what starts to happen is the matrix between them starts to harden. It starts to calcify a little bit. So notice our matrix in this area thins and hardens. And these cells have gotten really big and are really metabolically active. But if there's not a lot of matrix around them and the matrix around them is hardening, is it going to be easy to get the oxygen and the nutrients to these cells that they need? Is it going to be easy for these cells to get rid of their wastes?
No.
No. So the problem with this is these highly active big cells cannot get oxygen and nutrients. They can't get rid of waste. So what do you think happens to these cells?
They get
Yeah. The chondrocytes die. And when these chondrocytes die, they basically leave this hollow, hardened matrix void in the center. And does life like voids?
no.
No. So as a result of this, this space is a perfect environment to bring new cells, to bring new chemical signals, to change the growth processes that are taking place. And how do we bring new cells and new chemical signals?
They're blood vessels.
Blood vessel, exactly. And here is where now the nutrient artery grows into the diaphysis. Hopefully, one of the things that you will have the opportunity to do, and again, it is completely volunteer, it's not required, but a really good way to learn the bones is to actually hold them in your hands. So if you are comfortable and capable and willing and able to do it, go into the open lab in person on Fridays, and you'll get to actually hold and feel and taste, don't taste, the real bones that are there. And one of the things that you'll notice when you look at the real bones is every single long bone that is a real bone is going to have a hole in the center of the diaphysis. And that hole, the tiny little hole in the center of the diaphysis, is where that nutrient artery grows in.
Would that be like a perforating canal?
Yes, essentially it would be a very, very large perforating canal. That's a good way of thinking of it. So we started with a periosteal artery growing in. Now we have the nutrient artery that is growing in. And this nutrient artery, as we just finished talking about, is going to bring new chemical signals. It is going to bring new cells to this environment.
Does that artery form the hole, or how is the hole there?
It basically, the short version is a structure called a periosteal bud forms and penetrates into the surface. It's a little bit more complicated than we need to worry about. So just know that it grows into that area. Remember, everything we talk about in this class, the sky is blue. Every single concept we talk about is more complicated than we are going into depth on it. But so the simple answer for us, the sky is blue answer for us, is that it grows in. We don't need to know the mechanism of the budding that causes and allows that to occur.
Okay.
All right. That nutrient already grows into the diaphysis, brings new cells. And in particular, the new cells that it brings are osteoblasts. And as it brings osteoblasts into this area, we start to develop bone. Oops, well, it doesn't matter. No, that does. So we start to get the formation of bone here in this region. Now, let's think about this. When this bone is forming in this hardened, calcified cavity, do you think it's going to be perfectly arranged osteons with those nice concentric circles, or is it going to be a whole bunch of irregular points and processes and all sorts of different locations?
Irregular points and processes.
Yeah, exactly.
So is it forming like a spongy bone?
Exactly. So what happens here is that we form a small chunk of spongy bone. And as we know, our anatomists love to name everything. So they give this a big fancy name. And the big fancy name is the primary ossification center. Ossification center, this is the central region where bone is being formed. And why is this the primary?
So it's where it starts?
Yeah, because it's the first one that's occurring inside the bone. So we get this formation of this primary ossification, this first chunk of bone that is forming inside of the cartilage. All right. Now, is it going to be satisfied with just a single chunk of bone just at the center?
No.
No. So this primary ossification center is going to expand. And so from here, we are going to continue to produce more and more spongy bone, filling up our diaphysis. All right. Questions on that? All right. We have an issue though. Is this what we want our finished bone to look like? Just
No,
this
it
big,
should
huge
be
chunk
in
of
the
spongy bone?
Epiphysis, so it needs to push up somehow.
I'm sorry, say that again?
It needs to be in the in the heads of the bone.
True, but even the diaphysis, does the diaphysis look the way like we want the diaphysis to look?
It doesn't have the medullary cavity yet.
Right, exactly. So while this is a good start, this is not what we want our diaphysis to look like. So basically what we need to do now is we need to remodel the diaphysis. Or as you pointed out, we need to make the medullary cavity. Now, the problem we have is it's filled with bone, and there's just no way to get rid of bone. Is there a way to get rid of bone?
Don't osteoclasts kind of break down?
Bingo. So osteoclasts are going to form in the cavity, or in the diaphysis, let's say it that way. And you are absolutely correct. What those osteoclasts do is they start breaking down. Well, actually, let's cheat and do it this way. They start breaking down that bone matrix, hollowing the space out. And as they hollow the space out, are we going to just leave this as a hollow space?
No.
No.
No. What are we going to want to fill it with?
Endosteum. It needs like a layer.
Absolutely. We want to
make
form a medullary cavity. So let's do this. We remove most of the spongy bone. Notice not all. We leave a little bit of spongy bone on the ends. Forming the medullary cavity. You are then right. We're going to line the medullary cavity with the endosteum. And we're going to fill the space with what?
Yellow blood marrow, or I mean yellow bone marrow.
Yellow for this fetus growing
No,
inside
it's
of our
red.
body?
the red bone marrow.
Red bone marrow. Excellent. Perfect. So we are going to, and let's make it pink. Line that little bit of spongy bone that's still here lining the inner surface with that endosteum. And we are going to fill this space with red bone marrow. Excellent. At the same time this is going on, we need to form a thick layer of compact bone on the outer surface. So we're going to start producing, thick layers of compact bone on the outer surface. Thin little bit of spongy bone in the middle and red bone marrow filling the inside. At this point, hold on. At this point, now does our diaphysis look like a fully mature bone?
No.
The diaphysis.
Oh, the diaphysis, yes.
Yes. You're correct. The diaphysis is complete. You're right. The bone doesn't look like a fully formed bone, but at this point, our diaphysis is complete. All right. Questions on that? All right. But as you have smartly pointed out, nothing has happened in the epiphysis yet.
I have a question.
Yes.
So in the medullary cavity, it's technically supposed to have yellow bone marrow. So
No.
does that?
Remember, as an infant, as an embryo, we always start with all of our bone marrow being red bone marrow. As we age and mature, more and more adipose is deposited into these cavities so that as a mature adult, our diaphyses are filled with yellow bone marrow. Again, it's another one of those ways if you happen to stumble across a bone just in the middle of the woods and cut it open and look on the inside, you can get a relative idea of the age by looking at the composition of the bone marrow.
So we're basically looking at like an infant's bone then?
Yes,
Because
again, remember, this process is happening in utero. Remember, this
Right,
starts at 6 or 7 weeks.
Okay.
Right. So this is how long is baby in mom's tummy for?
9 months.
Well, yeah, 40 weeks,
Yeah.
right? About 40 weeks. So it starts at week 6 or 7. So this process is occurring. It's not till you reach adulthood that you have to start worrying about yellow bone marrow. So red bone marrow as an adolescent and definitely as an infant. Okay.
Thank you.
Yep. All right. So we have accomplished our goal of making the diaphysis look the way we want it. but nothing has happened to the epiphysis yet. And nothing will happen to the epiphysis for a while. In fact, the epiphyses.
I have a quick question.
Yes.
Even as adults, do we have a thin layer of spongy bone in our long
Yes.
bones? Because
Yes.
I found some didn't show it like in diagrams, so I wasn't sure.
It's a thin layer. So again, especially when you're looking at, so illustrations will typically show it, but like for instance, when we looked at that picture of the, well, here, let's do this. Notice here, it's not the best view, but it was one that I knew we had in our lecture, so it would be easy to see. Notice as we look in the epiphysis, as you expect in the epiphysis, it is filled with spongy bone, right? Entirely filled. However, if you notice looking in the diaphysis, you can see that thin layer of spongy bone that is lining, here now I'll erase that so we can see it more clearly. You can see that thin layer of spongy bone that is lining the diaphysis as well. So it is a thin layer, but there is a little bit in there.
Okay, so
Okay.
like would you like point to that and ask if there's spongy bone in like on an exam or?
No, I don't know why I would do that. I mean, I guess if you were describing the anatomy of a long bone, you would need to say that the diaphysis is lined with a thin layer of spongy bone. But on the exam, if I want you to identify a tissue type, I'm going to point here and ask you to identify this tissue and to be specific. And then what's your answer to that going to be?
Spongy bone.
Yeah. So, right. Whereas I can instead put that there. And what would you say to that?
Compact bone.
Yeah. Okay. All righty. Again, this process starts early, 6 or 7 weeks. However, the epiphyses aren't developed till much, much later. They actually develop right before birth, or in some cases, immediately after birth. All right. Now, I have run out of space here on my sheet. You do not run out of space when you're writing these exams, so you may not do that. In fact, I'll change colors to emphasize this point. Starts with the exact same process that we just finished talking about. Our chondrocytes are going to enlarge. As they enlarge, they're going to thin and harden the matrix around themselves. And as they thin and harden the matrix around themselves, what happens to the chondrocytes?
They die.
Excellent. Now we have a cavity. So what's going to happen with that cavity?
It needs nutrients or
Yeah, and so how are we going to bring them in there?
via blood vessels.
Exactly. A blood vessel grows into this environment. And in this case, this blood vessel is called the metaphyseal blood vessel. So that metaphyseal blood vessel grows into the environment and it brings new chemical signals, it brings new cells. And so as a result of that, we get the formation of a chunk of spongy bone. And what would we call this chunk of spongy bone?
Trabeculae? Or would it just be?
True, it is. These are indeed trabeculae because that is what makes spongy bone. But remember, the first chunk of spongy bone that formed we called the primary ossification center. So what do you think we call this one?
Mary?
And notice we have one diaphysis. So we have one primary ossification that happens first. Our secondary ossification center happens second, but it happens in the epiphyses. And how many epiphyses do we have?
2.
2. So we form 2 secondary ossification centers, one in each epiphysis.
So it starts with the same process for one and
Yes.
like
So
steps
again,
1 and 2.
if you were describing this on the exam for the second, for this, so at birth or right before birth, right? Then if you think about, if we look back at what we saw, what happens is, what happened? Right, so here.
So that we would need to specify.
Yeah, you would describe the process.
Again, okay.
Right? Chondrocytes grow in size, matrix thins and hardens, chondrocytes die, right? Metaphyseal artery grows in to the epiphysis, right?
Do we need to specify?
And
proximal
forms the
mandible?
secondary ossification center. Again, I would have written all of that out. I just don't have room here at the bottom. But if
And
you
just
were
for-
writing this out as an essay question, and I guarantee this is one of the essay questions on your exam. Now it's in a bank, so I don't know who of you will get it, but I guarantee some of you, we're going to get this as an essay question. So don't just say the same process occurs. That's not descriptive. That's not explaining. Explain it again. Tell me what happens here. Because while it starts the same, it's not going to finish the same. There are going to be 2 big differences, really 3 big differences. One big difference that we already talked about is the timing, right? One of these processes occurring at 6 weeks, where the other is occurring at birth. Yes, Amanda?
So are they happening in the distal and proximal at the same time?
Yes, this is happening in the distal and proximal at the same time.
And then do
This
we
is
specify? I guess we would need to specify that.
You need to say that it's happening in both epiphyses, yes.
Okay.
Right, but it's the same process in both epiphyses. So you don't have to explain it 3 times, just explain it twice.
Okay.
But as I mentioned, there's 3 big differences. One, we already talked about, the timing. This is occurring much, much later in the process. But let's talk about the other 2, right? As we saw, we made a primary ossification center and then we hollowed it out. Is that what we're going to do here? We're going to make a secondary ossification center and then we're going to hollow it out?
No.
No. So in the case here, we leave the spongy bone. We do not hollow it out. There's no medullary cavity in the epiphysis. Now, notice one other thing. In our diaphysis, we converted all of our cartilage to bone. Are we going to want to convert all of our cartilage to bone here on the epiphysis?
no.
No, I'm asking the question, so it should be a super obvious answer, but absolutely. We are actually going to leave hyaline cartilage in 2 locations. Where are the 2 locations where we're going to leave hyaline cartilage?
The articular
Or on the outside of
Perfect.
outside, and
So
then the
on the outer surface, we're going to leave it as articular cartilage. So hold on,
On the
let's label
epiphysis.
that.
and then our growth plate, epiphyseal growth plate.
Yep. So we have that articular cartilage. So we're going to leave some of it out there. And what's the function of that articular cartilage again?
To join our bones.
Yeah, to form the joint. Excellent. And we're going to leave some hyaline cartilage here in the center. And what did you guys call that?
epiphyseal.
Excellent. And what's its function?
To allow the bone to grow in length.
Exactly. This is where we're going to grow the bone in length. And with that, we have now accomplished our goal. We have made a long bone. We have taken a chunk of cartilage and converted it into a long bone. All right. Questions on this?
So stepwise, are we allowed to just put this all under 4, or do you want us to break it up?
As I've said before, I don't care how many steps you give it to me. I think your book uses 6 steps or something like that. We'll look at that. We'll go over the process there. You can do 2 steps. You can do 17 steps. I don't care how many steps you use, as long as you give me all of the information. I want all of the information in order. How you break it up to make sense of you is perfectly fine with you. Right? Because I'm sure if you look in 5 different textbooks, they'll probably break it up 3 different ways. Not everybody's gonna, but everybody has the same information. So how you break up the information is completely up to you. I don't care. Just make sure that you have all the information there. Okay. All right. I know we normally go over this immediately right again, but I think we went a little long. So let's go ahead and take our first break. Give this a chance to soak in. And then after this has had a chance to soak in, we'll come back and we will go through the entire process again with the pretty picture from your textbook. So let's go ahead and take our first break. It is 9:21, so let's come back at 9:36. And at 9:36, I will start the recording again, and we will look at endochondral ossification with all the pretty pictures and all the pretty words from your textbook. All right, questions on any of that? All right, I will see you in 15 minutes. All righty. We have just finished talking about endochondral ossification on the board. Let's look at the pretty pictures from your textbook that go through the same process. And we, of course, got to get back to where we were before. Here we go. Excellent. So, as I mentioned, in this process of endochondral ossification, our goal is to convert cartilage into bone. As we mentioned, this is how we make all the long bones. And that's what we see here on this illustration. Notice here you see both sides of it. And again, remember, as we talked about, this starts around week 6 or 7 weeks of development in utero. As we talked about, we have this chunk of cartilage that is growing, but we want it to make something else. So we need to change the chemical signals. We need to change the chemical cues, bring new chemical signals in here. And how do we do that again?
Blood vessels.
Exactly. So a blood vessel grows into the perichondrium. As a result of that, we start to get osteoblasts that form. And an osteoblast is going to do what an osteoblast is going to do. It is going to make bone. And so we start to get the formation of this thin bone cuff or bone collar that forms on the outer surface. And because that perichondrium is now sitting on top of bone, we no longer consider it a perichondrium. What do we consider it now?
Periosteum.
Periosteum. Excellent. All right. Questions on that? All right. Remember, as that is happening on the outside, changes are occurring on the inside as well. Our chondrocytes are becoming overactive and enlarging. They're getting big and they're swelling, which isn't a big deal in and of itself. But as these large chondrocytes start to form in the center of our bone, the matrix around it thins and it calcifies. And so as a result of that, those chondrocytes die. And that leaves this cavity in the center. This cavity is the perfect environment for some changes to take place. But to get those changes to take place, what do we have to do?
fill it with the endosteum,
True. And how are we going to get that endosteum into there?
the blood vessels,
Yeah, blood vessel.
nutrient artery.
Exactly. So that exactly the blood vessel, in this case, the nutrient artery, penetrates into the center of the bone, and osteoblasts start to form. And osteoblasts start to make matrix. And again, they're not going to make a perfectly aligned, uniform layers of matrix. There's going to be all these irregular shaped layers of matrix. And so we are going to get a big chunk of spongy bone that forms. And what do we call this chunk of spongy bone that forms in the center of our diaphysis?
The red bone marrow?
Nope.
The
spongy
primary
bone.
ossification center.
Exactly. It is our primary ossification center. You're right. We're going to bring bone marrow in there, but not first. First, the bone forms first. And that chunk of spongy bone we call the primary ossification center. And remember that primary ossification center is not going to just stay in the middle. It's going to start to expand to fill the entire diaphysis with spongy bone. Again, it's fine. We're converting into bone, but that's not what we want our mature bone to look like. We don't want our mature bone to just be spongy bone throughout the entire diaphysis. So we have to modify it. We have to make it look like the mature bone. And one of the primary ways we do that is by hollowing out, removing most of that spongy bone. And how are we going to remove most of that spongy bone?
Osteoclasts.
Exactly, right? Osteoclasts are going to come in and remove most, not all. See, there's still a little bit of spongy bone lining the surface, but most of that spongy bone forming the medullary cavity, which we fill with jelly beans, with red bone marrow, filling it filled with that red bone marrow that fills the space. And at the same time, we are organizing and thickening that outer layer, forming a thick layer of compact bone on the outer surface. And lo and behold, compact bone outer surface, thin layer spongy bone, red bone marrow in the center, filling that medullary cavity. Our diaphysis is now complete.
Is the endosteum, I guess, on the inside of the spongy bone?
Well, it's not inside the spongy bone, but it is
Right.
lining the surface of the spongy bones.
Okay.
So yeah, so it's not in it, but
Right.
it's lining it. Yes. So
Okay.
it lines that medullary cavity. It lines that surface. Amanda, you had a question as well?
Yes. Is there, does the, when it becomes a periosteum, does it still have mesenchymal cells?
Yeah. Again, notice nothing about the perichondrium changes.
Okay.
Still a dense irregular connective tissue. It's still housing immature cells. The only thing that changes is what it's sitting on. Before it was sitting on cartilage, and now it's sitting on bone. So it hasn't changed at all, except the one thing that has changed, you could argue, is its location. Before it was sitting on cartilage, now it's sitting on bone. And since we name it based on its location, and its location has changed, that's why we change its name. It's not any different from a serous membrane. A serous membrane that is on the heart is the visceral pericardium. If we peeled that off and put it on a microscope slide, and we took a parietal peritoneum and peeled it off and looked at it under a microscope slide, those two tissues would look identical. They're both serous membranes, simple squamous epithelial tissue called a mesothelium, sitting on top of an areolar connective tissue. They're identical. The only difference is where they're found, their location. One is on the surface of the heart, one is lining the abdominal pelvic cavity. So many of our names based on location are really just that. Names we give things because when they're in different locations, they have different names. All right. Again, all of this started around week 6 or week 7, and so this finishes pretty early in the developmental process. right? When you're getting that ultrasound at week 20, you can start to see the ribs and the vertebrae and all those kind of fun things inside of your growing fetus inside of mom's belly. However, when baby's born, and again, I'm not encouraging you to do this, but has anybody ever bent the fingers of a newborn baby?
No.
They're like super bendy flexible. And why are they super bendy flexible? Because their epiphyses haven't formed yet. The epiphyses of the fingers of the digits don't actually form till after birth. So while this happens, the diaphyses occur very, very early. The development of the secondary ossification centers, and notice here you can see it's occurring in both epiphyses at the same time, happens much, much later, at the time of birth, or even immediately after birth, shortly after birth. Again, I'm for brevity putting this here, but don't write it this way on the exam. Make sure you explain the process. This is similar to what we saw in the diaphysis. Chondrocytes grow big, chondrocytes die, leaving a cavity. When they leave that cavity, a blood vessel grows in. This time it's the metaphyseal. Notice it's not going to grow in like our picture is drawing it here straight from the side. It'll draw in more from the neck and like that, bringing new cells, bringing new nutrients, and forming a chunk of spongy bone. And of course, that chunk of spongy bone we call the secondary ossification center. And again, for me, the way it's easy for me to remember this is primary occurs first, secondary occurs second, right? So that's easy. But also the primary occurs in the diaphysis, and we only get one of them, whereas the secondary occur in the epiphyses. So two means second, but there's also two secondary ossification centers. So that helps it make it easy for me to remember. So the process starts the same way. We form that chunk of spongy bone in the center of the epiphysis using the exact same method, because as we said, life is lazy, using that exact same method that we did for the diaphysis. But there are two, really three big differences. Difference one is the timing. Difference two is that we don't hollow out the spongy bone. We leave the spongy bone. So the head, the epiphysis contains spongy bone. And what was the third difference?
We leave the hyaline cartilage in two locations, which would be on the...
Exactly. We don't get rid of all of the hyaline cartilage. Hyaline cartilage is going to remain in two locations. We are going to leave some of it up here on the epiphysis for forming our joint, and we're going to call that the articular cartilage. And we are going to leave some of it in the metaphysis, where it becomes the epiphyseal plate, which we're going to use to grow the bone in length. And with that, we have now used endochondral ossification to make a long bone. Questions on that?
So with the possible essay questions, would this just be one possible essay question, or could you ask us like, what is the difference between the primary and secondary ossification? Like, because there's technically two stages almost.
I think something more discrete like that lends itself more to like a multiple choice question or something along those lines. However, like I said, I guarantee with 100% certainty, describe the process of endochondral ossification is an essay question in your test bank. So yeah. All right. All right, excellent. So last point I want to emphasize is notice in reality, this whole process of converting cartilage to bone is really the same process happening 3 times. Notice a blood vessel grew into the environment. We started making bone, right? First, we did that in the periosteum. Then we did that in the diaphysis. Then we did that in the epiphysis. A blood vessel grew in, changed the environment, changed the chemical signals, changed the cells that were present, and we made bone as a result of it. And as we talked about, one of the biggest differences between cartilage and bone is very well vascularized. And notice also that while it starts with these three distinctly different blood vessels, one that grows into the periosteum, one that grows into the diaphysis, one that grows into the epiphysis, do those blood supplies stay separate from each other?
No.
No, as you can see, they all grow together. Remember, bone is an incredibly dynamic tissue. It is incredibly well vascularized, which is why it's able to heal so quickly. And notice, while your book here emphasizes all these blood vessels going to all these locations, everywhere these blood vessels go, what else follows along with them?
Artery or
Sure, we got
vein.
arteries and veins, but what travels alongside those arteries and veins?
Nerves.
Nerves, and one other thing. No, close though. Lymphatic vessels. Right, lymphatic vessels, absolutely. So again, our lymphatic vessels and our nerves are going to follow these blood vessels, all becoming interconnected, giving that huge dynamic process to the tissue. All right, so that is our long bone. Any questions on that? All right, let's talk then about our second bone formation process, intramembranous ossification. And again, let's start on the whiteboard. For this one, again, just watch, look, think, listen, and let's go over this process together. So again, this is intramembranous ossification. Oops, no, that's all right. What type of tissue are we converting to bone here? It's not cartilage, so what was left? We're going to have this fibrous connective tissue membrane, let's say it that way, that we are going to convert into bone. Now, remember, we mentioned we use the endochondral ossification for most bones. This one is primarily used for flat bones of the skull and the mandible, and a couple other bones are done this way as well. Once again, we need a starting point. And that starting point is going to be that fibrous connective tissue membrane. So we're going to have this, what is going to look very much like a dense irregular connective tissue. Again, embryonic tissues are not identical to our mature tissues, but it looks close enough to a dense irregular connective tissue. And within this dense irregular connective tissue, we're going to have some mesenchymal cells. So these are mesenchymal cells. That's way too big. And our collagen fibers. All right, much simpler starting point. All right, once again, we are going to start with a chemical change. And that chemical change is going to cause these mesenchymal cells to start to produce osteoblasts. So again, we're going to get these chemical changes that take place. And as a result of that, our mesenchymal cells produce a cluster of osteoblasts. And what do you think osteoblasts do?
They're immature cells that make bone cells.
Yeah, they're immature cells that their job is to make bone matrix. And so that's what they're going to do. They're going to make a little bit of bone matrix and surround themselves with this matrix. And suddenly I'm going to have this little cluster of spongy bone that is forming here within the center of this tissue. And what do you think I would call this cluster of bone that forms in the center of this tissue?
Maybe an osteum? Is that how they form?
Well, remember, it's forming more than one. It's not quite organized yet. So it's spongy bone. So it's forming a cluster of spongy bone.
So
Didn't we
trabeculae?
just see 3 clusters of spongy bone in the last process?
That's an ossification center.
Yeah, it forms an ossification center. Now, remember before we talked about primary and secondary ossification centers. Notice I didn't give this one a name like primary or secondary. Because do you think this is the only cluster that's forming?
No. it's
No.
putting in
No, another
all-
cluster may be forming over here, and another cluster may be forming over here, and another one over here, and another one over here, and another one over here. And are they necessarily sequential in the way they form?
No.
No. So in that case, I'm not going to bother primary, secondary, tertiary, quaternary, and so on and so forth. We just know that many of these ossification centers are going to form around the same time. And since there's many of them, we're not going to worry about trying to give them names. We're just going to call them generic ossification centers, little chunks of spongy bone that are forming throughout this membrane. Now, once I form a tiny little chunk of spongy bone, am I going to be content with that and just leave it as that tiny amount and stop the process at that point?
No.
No.
No. So what's going to happen is that these ossification centers are going to expand. And as they expand, they grow into each other. And as they expand and grow into each other, they form our trabeculae. These are regular points and processes of our spongy bone. So this one's going to expand outward. And as it expands outward this way, and then this one expands this way, and this one comes out this way, and this connects here, and this connects here, and this one's connecting here, and that's connecting like that there. We're going to get the formation of all of these trabeculae that are going to form as our spongy bone forms these trabeculae and expands. All right, now notice as the tissue expands, 2 things are going to occur. The first thing that is going to occur, is notice as this tissue is expanding outward, and I'll cheat and change the color slightly. As it is expanding outward, it is pushing on the collagen fibers. So there's this mesh of collagen fibers. And as the tissue expands, it basically condenses these collagen fibers along the outer surface. Now, are these collagen fibers going to be perfectly arranged as that tissue expands around the outer surface?
No.
No. So notice we're going to start to get this formation of a dense, irregular connective tissue that forms on the outside of this bone. And notice as it's forming on the outside of this bone, it's going to capture those mesenchymal cells underneath it. Well, look at that. I have a dense, irregular connective tissue on the outside of my forming bone, and it has mesenchymal cells inside of that. What does that sound exactly like?
It sounds like the endochondral ossification.
Well, okay, sort of, but what structure in particular is a dense irregular connective tissue that houses immature cells and is on the outside of bones?
Is it the circumferential lamb?
Yeah, Mitch has got it. It's the periosteum, absolutely.
Yeah.
As it condenses down on the outside, notice it ends up forming the periosteum on the outer layer. So this dense irregular connective tissue that forms becomes the periosteum on its outer surface. Now, notice one other thing as well. There's all these spaces, this cavity inside of the trabeculae. And as we mentioned, does life like cavities? Does life like empty spaces?
No.
No. So what would be convenient to grow in there and deposit into these spaces?
Blood vessels.
Exactly. Blood vessels are going to grow into this area. And as they grow into this area, they're going to deposit what? What do you think we're going to fill these? Yeah, exactly. It's going to fill this space with bone marrow. It's going to line this tissue with a periosteum. So we're going to get a periosteum that, an endosteum that forms on the inside. So that endosteum is going to form. And when we form this endosteum, we are then going to fill these spaces with red bone marrow. So the second thing that happens is this tissue expands, is blood vessels grow into the spaces between the trabeculae. They form the endosteum and fill the space with bone marrow. Oops. Excellent. All right. So far, so good?
Yep.
All right. Now, notice, now that our periosteum has formed on the outer surface, now we're able to start producing, and actually, let's do it this way. We're able to start producing a thick layer of more organized bone on the outer surface. And this, well, really thin layer of compact bone on the outer surface. Again, remember, this is forming flat bones and irregular bones and stuff like this. So we form a thin layer of compact bone. And what would we call that thin layer of compact bone on the outer surface?
The cortex.
Yeah. Our cortex forms. And notice at this point, we pretty much have the anatomy of the bone that we want. Yes? Yeah. With one exception. What are the chances that these clusters of ossification centers formed in just precisely the exact location to give us the exact shape of the bone we want? What do you think? Good? Good chance? 75%? 90%? That it's going to be perfectly aligned to form the exact shape of the bone we need for our mandible?
No.
No.
No. Absolutely not. It's not. So the last step, the last thing we have to do, now we have a bone-shaped bone, but now we need to make it the shape we want. So the last step is going to be remodeling. Right? So let's say, for instance, we needed to add some bone in this area down here and expand this out. Are we capable of making cells that can make more bone and expand the tissue out?
Yes.
Yeah, and what kind of cells would be useful for that?
Mesenchymal.
Mesenchymal.
Well, but which ones actually make the bone?
Osteoblasts.
Right, we can make osteoblasts to add bone to the areas that we need it. On the other hand, if we needed to, oh, I don't know, remove bone in an area, do we have cells that would allow us to remove that bone in the area?
Osteoclasts.
Yeah. And so using osteoblasts and osteoclasts, we can now take this bone and make it the perfect exact shape that we want, forming this bone into that mandible or into that flat bone of the skull.
So that's how they get the specific shape, is using osteoblasts and osteoclasts to mold.
Yep, to remodel it into the shape that we want. Absolutely. Questions on that? All right, we've done it here. Again, let's look at the pretty pictures from your textbook. Notice, as we mentioned, here we are inside of that embryonic membrane. We see our collagen fibers. We see a bunch of mesenchymal cells. And notice we get this clustering together of osteoblasts that are going to form. Now we're just looking at one location, but remember, this is going to be happening in many locations inside of our fibrous connective tissue. And they start depositing that calcium crystals onto the collagen fiber, and they make some bone matrix. Of course, when a cell surrounds itself with matrix, we know it becomes an osteocyte. And notice it has its little processes now going out into those canaliculi. And so we have a little clump of bone that is forming. However, we have many clumps of bone that are forming. And these many clumps are going to grow into each other. And as these many ossification centers, and let's emphasize that here, as they expand into each other and grow into each other, notice they're forming those irregular points and processes that are the trabeculae of our spongy bone. And notice here, two things are going to occur while this trabeculae is forming. For starters, there's the cavity in here. And as those cavities, those spaces between the trabeculae form, blood vessels are going to grow into these spaces, filling it with red bone marrow. But notice also, they haven't shown us the fibers. Notice there would be fibers out here as well that are condensing down on the outer surface. But what they have done a nice job of showing is the mesenchymal cells that are being condensed down on top. And as those fibers and mesenchymal cells are condensed down on top, our periosteum forms. The nice thing about that periosteum is it allows for the organized production of bone. And with that organized production of bone, we can form a thin layer of compact bone on the outer surface, which of course, as we mentioned, we call our cortex. Oops. And then using osteoblasts and osteoclasts, we can remodel the bone to give it the precise shape that we want. Yes, Amanda.
Why doesn't the compact bone in this like get a medullary cavity? Like what stops it from, you know, like a long bone has that process? What makes that not happen in the compact bone and in the spongy bone?
Well, essentially what's happened is, especially here, you're making the spongy bone first, and then you're just putting layers of compact bone on the outer surface. So that's what's happening here with this process. In the other process, in the endochondral, if you think about it, they meet in the middle. We start a thin layer of compact bone, that bone collar on the outer surface, that grows thicker and thicker and thicker. But at the inside, we send in that bone, and we form the primary ossification center. So you've got compact bone growing in, spongy bone growing out, and then they meet. So it is slightly different in the process there, but in both cases, the spongy bone in the center stops the compact bone from continuing to grow inward.
Okay.
All right. Questions on this second bone forming process?
I do have a question.
Yes.
Sorry. So for the intramembranous ossification, it's specifically for flat bones?
Yeah, flat bones, then the flat bones of the skull, and most of the flat bones of the skull, the mandible, a couple other bones.
Okay. And so then the endochondral ossifications for like long bones, short bones, all of those types, are just long bones?
Most of the bones of the body are formed using endochondral ossification.
Okay.
And most, including all of the long bones. So all of the long bones, but most of the other bones as well use it as well. So there are just a few bones mostly associated with the skull. There are a couple outside of the skull, but most of the bones of the skull use this process, the intramembranous, but everything else pretty much uses endochondral.
Great, thank you.
Yep. Any other questions? All right, so we have two processes for making bones. And someone remind me again, how many essay questions does that give us?
3.
3. What was the third process then? The
How
third
they're
essay question?
Compare.
Again, one of the issues that we had on this last exam is there are a couple questions where people were asked to compare things, and they just described the two processes. Is that what we're doing when we're comparing things?
No.
No. Again, you are not required to set it up this way, but sometimes I feel that it is useful, at least while you're thinking about this, to remind yourself of the question. Is that when you're going to compare endochondral to intramembranous, use a third column. And that third column is the characteristic that you are comparing. This allows you to make sure that you are comparing apples to apples, right? Because telling me that epithelial tissues are cell dense and connective tissues are deep, while both of those are accurate statements, are they a comparison of the two?
No.
No, right? Saying epithelial tissues are superficial and connective tissues are deep, that is a comparison. Saying epithelial tissues are cell dense and connective tissues have fewer cells and have that matrix that contains fibers and ground substance, that is a direct comparison. So make sure you're comparing the two. So let's do that. Tell me a way that endochondrial and intramembranous ossifications are different from each other.
Types of bones that form.
Excellent. Endochondrial forms what types of bones?
Long bones.
Most
True,
of
and again, most bones, but
yeah.
definitely all long bones. Whereas intramembranous primarily produces
Flat bones. And like the mandible, flat.
Yeah.
The skull.
excellent. What's another way they're different?
Is timing one?
Yeah, timing can be. What do we know about the timing with endochondrial ossification?
6 to
It happens
7 weeks.
early, 6 to 7 weeks. Fetal.
Starts early, but what else? Is it a continuous process?
Yes.
Yes?
No.
No. It's not a continuous process. We make the diaphysis and then we wait, in some cases, months until birth. And then it finishes
Oh, I see.
just before, or even in some cases after birth. All right. But what about intramembranous ossification? Is there a big gap in that? I didn't mention one, so what do you guess?
No.
No. So this one basically starts early, because all bone making starts early, but is a continuous process. So it's going to finish early as well. Excellent. What's another difference?
Can you talk? I know we said types of bones, but can we talk about like the compact and spongy?
Well, but do both have compact and spongy?
Oh, they both have. Okay.
Yeah. Is there characteristics that both don't have?
Structure.
How so? Be specific.
Trabeculae versus osteum.
Well, but again, remember, both have compact bone and both have spongy bone. You're comparing compact and spongy bone now. That's not what we want. We want to compare endochondrial ossification, intramembranous ossification. So how are those different? Oh, here we go. Excellent. And not just their specific location, but we can even go simpler than that. How about just simply the number of ossification centers? How many ossification centers does endochondrial ossification have?
3.
3.
How many does intramembranous have?
Several.
Many.
Many, absolutely. And the timing of them, right? Here, there's first primary, and then again, way later, we have 2 secondary. Whereas here, we have many, and they all form at the same time. Excellent. Give me another one. I can think of at least 2 more really big differences.
Their location. So
What do you
one's like
The one
ones with cartilage.
One's appendages.
Okay, true, but I think that goes along with the type of bones that they are.
Okay.
So I think that, although I do like what Ava was saying, your starting point definitely is different,
Yeah.
right? The starting point for endochondrial, what are you starting with endochondrial ossification?
A little block of cartilage.
Hyaline cartilage.
Island cartilage. Whereas what is your starting point with intramembranous?
Osteoblasts.
No, what type of tissue is it starting in?
Oh,
Connective tissue.
Starting- Maybe instead of starting point, we say starting tissue.
Fibrous.
Maybe that helps. Excellent. Fibrous connective tissue. Excellent. But that's an excellent one. It wasn't even one of the 2 I was thinking of. I like that one. That was
Sorry.
excellent. No, don't be sorry. You gave us more information. That's awesome. It was absolutely correct. Right? Let's think of 2 other ways. Right? Blood vessels. are a huge feature in this process. But let's think of it this way. With the endochondral ossification, which occurs first? Do the blood vessels grow in, or do we form bone first?
Blood vessels are first.
Yeah, blood vessels grow in, and then we form bone. Is that what happened in the intramembranous ossification?
No.
No. Here, notice we formed the bone first, and then once that bone formed, and then the blood vessels grew in. And also, what about the periosteum? What was there first? The dense irregular connective tissue that becomes the periosteum and then bone, or bone first and then the periosteum? Which is which in the endochondrial?
I think bone first.
Well, but be careful. We had a dense irregular connective tissue. We didn't call it a periosteum. It was a perichondrium.
Oh, okay.
But that perichondrium was there first. It then became the periosteum. All right. So maybe we'll put perichondrium in parentheses. Because again, remember, it was the exact same thing. So the perichondrium, the periosteum was there first, and then the bone formed. But it's a lot more obvious with intramembranous. Which formed first in the intramembranous?
The bone.
Yeah. Excellent. So, and I'm sure there's probably a handful more we could think of as well. But notice again, while you're not required to organize it this way, I find that giving yourself that characteristic really helps you to make sure you're comparing apples to apples and oranges to oranges when we're comparing these two processes. All right. Excellent. Questions on that? All righty, spectacular. Well, the good news is that's all we're doing for lecture today. Whoops, need to come back here. So what we are going to do next is we're going to take a break. But before we take a break, I want to do a quick introduction. During the break, I will break you into your groups so that the groups that are presenting today will have the opportunity to meet and get set up and get ready for their presentations. So I'll break you up into your groups to be able to do that. But before we do that, yes, Amanda, you had a question?
Are you enabling screen sharing for?
Yes, I will, and in fact, I'll do that right now, even though you shouldn't have to do it now, but that's a great point. So let's, oops, wrong button. Advanced. All participants can share. So yes, that is now set up. Perfect. So we'll be able to do that. But before we do that, let me do my part of the introduction. Our goal is to talk about the skeleton. And the skeleton is how many bones again?
206.
Name bones.
Named bone.
Named bones. Excellent. Excellent. 206 named bones. And it's basically divided into two parts, the axial skeleton and the appendicular skeleton. What are the structures of the axial skeleton?
The skull.
Skull?
Vertebrae?
Yep, vertebral column, the vertebrae, and no, not the muscles. Rib cage, there you go, the bony thorax, absolutely. So it is the skull, it is the bony thorax, and the vertebral column. And of our 206 bones, it makes up 80 of the named bones that we are responsible for. As I mentioned, our goal today is to hopefully make some sense of the skull. So let's start with the skull. The skull is comprised of 22 total bones, and those 22 total bones are divided into one of two types. And let's cheat and go to our whiteboard for this. 22 bones to the skull, and those 22 bones are divided into two main types. What are the two main types of bones that make up the skull?
The cranial bones and the facial bones.
Cranial bones and the facial bones. Excellent. Just out of curiosity, how many cranial bones do we have?
8.
8. How many facial?
14.
14. There you go. That equals 22. Perfect. Excellent. Name for me the 8 cranial bones. Give me them one at a time.
Frontal.
Frontal.
Temporal.
What else? I'm sorry.
Temporal.
Temporal.
Occipital. Parietal. Ethmoid. What is it? Sphenoid or something?
Sphenoid.
Sphenoid.
There you go. Excellent. And that is 8 bones. Wait, hold on. It's only 6 bones. What are the other 2?
Well, the parietal and the temporal each have 2.
There you go. For the parietal, it is a paired bone, meaning that there is 2 of them. And the temporal bone is also a paired bone, meaning there's 2 of them. With the skull, it's pretty easy to tell whether something is going to be paired or not. If a bone is either on the midline or crosses the midline, then it's a single bone, right? I have this big, huge single 4-head bone, or for me, it's a 5-head bone, right? And that is that frontal bone here in the front, right? The ethmoid crosses the midline, the sphenoid crosses the midline. But what are the 2 bones that don't? The 2 lateral temporal and the 2 lateral parietal. So those are our paired bones. And of course, what do these 8 cranial bones do?
They just enclose your brain.
Yeah,
They protect your
they
brain.
form the cranial cavity, right? One of those cavities that we talked about. Its job is to protect the brain. Oops, not how you spell brain at all. There we go. So then what do the facial bones do?
Movement.
Do all these
Make up
bones
your-
move?
No.
No. So
They make your facial features.
yeah, they form the
Structure.
face.
Yeah.
Sometimes the obvious answer is the right answer. And it's one of those. Facial bones form your face. Excellent. So let's identify those. And now we know the trick. We know they're either going to be paired or unpaired. So let's say that right from the bat. Give me one of our facial bones.
Zygomatic, which is paired.
Excellent.
Maxillary, which is paired.
Times 2.
The mandible.
Mandible. 1 or
And
2?
that's 1.
1. Excellent. What else?
Palatine, which is paired.
Times 2.
Vomer, which is not paired. Nasal, and that's paired. And there's
2,
like an
4, 6,
inferior
8,
nasal.
10. A couple more. Say again.
Inferior nasal, like concha or something. I
Concha.
think that's concha. That's
But
paired.
again, I don't care how you paired or unpaired.
Paired.
So what does that leave us with?
Lacrimal.
Yep, there we go.
Which is paired.
Is paired.
Perfect. There you go. So those are the 14 bones of the face and the 22 bones of the skull. Notice again, if you look at it, there are only 2 unpaired facial bones, the mandible, which crosses the midline, and our vomer, which is actually dead on the midline. All the others are paired, which is, again, kind of one of those things that we know about the face. One of the things about the face, in fact, one of the features that most people tend to find attractive about individuals is, right, that symmetry, exactly. You have that symmetry of the 2 sides. So of those facial bones making up the face. Excellent. So most of those are paired because we have a left side of the face and the right side of the face. Now remember, all of these bones are bones you are required for identifying, and all of these are bones you're required for identifying on an intact skull. Right?
What does that mean?
Like I show you a skull, like the one that's back there,
Right.
and you're able to, there you go, like that one there. And you are able to identify. However, I'm not going to have like a nasal bone. And if you've seen the nasal bone, it's like a square chip of bone sitting on the table by itself, and you have to figure it out. However, remember, as we talked about in the last class, the temporal bone, the ethmoid bone, the sphenoid bone, the maxillary bone, and the mandibular bone, and the palatine bone. These 6 bones are all bones that you need to know disarticulated. So these could possibly be shown on as a bone sitting by themselves on the counter. So hopefully the people showing these bones today will have done that for us. There's one other special thing that we want to talk about. This mandible. Not only is it special because it's one of the bones that we need to know disarticulated, but what else is special about the mandible? Well, it's not the only one that has teeth. That is partially true, but the maxillary also has teeth. What is the one characteristic that the mandible only has? Let me rephrase that. What is the one thing the mandible does that nothing else does? What? Moves.
Moves.
Absolutely. Right? The mandible is the only bone of the skull that moves. You think my lectures are bad now. Just imagine what they would be like if I couldn't move my mandible. Right? So absolutely, it is the only movable bone of the skull.
So is the majority of our anatomy or our lab section going to be just the bones and the bone features?
It's going to be a large part of it, absolutely. However, remember, as we talked about, a bone feature is a characteristic of the bone itself. So for instance, every single bone on this list has at least 6 or more bone features on them. So of those 6 plus, how many are you responsible for the frontal bone?
1.
1. How many are you responsible for the vomer?
None.
None, exactly. Does that mean the vomer doesn't have bone features?
No.
No. So you are just responsible for the bone features on the list. However, remember, these bone features of these skull bones will come together to form skull features. A skull feature is a characteristic of the skull. So 2 or more bones have to come together to form those skull features. It's the same thing with the skeletal features, just the skull feature is a skeletal feature of the skull. For those, you need to ID the skull feature, but you also need to know the bones and or bone features that form them.
So with one question, you can ask 3 different things, basically.
Or yes, yeah, essentially yes. And we'll look at that when we're going through this. We will go through this together, okay? All right, so these are all the bones you're responsible for. We are now going to split up into our groups that are going to be responsible for presenting this information for us. So you're going to show us the bones and bone features that are on the list. Again, please be on camera. Please make sure that we can hear you. But other than that, like I said, if you have it written on a piece of paper and you want to read it on the piece of paper the whole time and you don't want to look at the screen the whole time, that is totally fine. Just make sure you're on camera. Notice no one's looking at you anyway. They're going to be looking at the pretty pictures that you're posting, showing us their bones and bone features. And again, you're responsible for these bones and bone features. You are not responsible for most of the functions. For instance, the jugular foramen. What do you think the jugular foramen is for? Yeah, the jugular vein passes through it. But that is not a question I'm going to ask you on the exam. Knowing that may help you, but I'm not going to ask you the function of most of these structures on the exam, unless its function is to form a joint. If, for instance, the trochlea of that humerus that we talked about is a bone feature, you need to know what it does. It forms a joint with the ulna. So things that form joints, that we need to know. But other than that, you don't need to know the functions. If knowing the function helps, knowing that the jugular vein goes through the jugular foramen helps you to remember it, winner. But I won't be asking that on the test. On the test, I'm not going to point at that hole and say what passes through this hole. But
So are joints what connects two bones together?
Yes.
Okay.
Yep.
So that's just what's classified as a joint.
Exactly.
Okay.
Yep. All righty. So with that in our pocket, let me go ahead and break us into our groups. So give me a second. I need to, all right, I'm gonna need to juggle some stuff here real fast. So I need to put that there and come here. And then go there. As I mentioned, I think it's the first three groups that all have bones they're responsible for today, bones of the skull. So those are going to be the three groups that are presenting today, but I'm going to put everybody into their groups just so, again, you guys can work and coordinate. We will then take our break and then also, and then come back. So once you're in your groups, if you want to go take your break first and then come back and study or prepare first and then take your break, you're welcome to do that. But let me
How long is the break?
I'll figure that as soon as I get the break room set up. Once I get the break room set up, then we'll figure that out. Where are my break rooms? Oh, they're there. Whoops, hold on. Alrighty. And I believe for this, we're going in order. So group one is the first group to go. I think you have the frontal bone, so you'll be the very first group that is presenting, and then we'll go from there. There you are. All right. Group four, you're missing somebody in your group, but they are not presenting today, so that shouldn't be an issue. Hopefully you've heard from them though and have coordinated your materials. Again, on the off chance someone doesn't show up when you guys are presenting, make sure everybody is prepared to present all of the materials. After all, you need to know all of it for the exam, so there's no reason not to be prepared ahead of time. And the last group. All righty. Our rooms are now set up. It is 10:38 right now. So tell you what, let's meet back here at 11. That gives you guys a chance to take a break, and it gives you a chance to coordinate. So at 11 o'clock, I will close up the rooms and we'll meet back together to go over the materials on this. So that gives you guys a chance to have a break, but also a chance to work in your groups as well. So I will see you here in 21 minutes. So that we can start our group presentation. So I'll see you guys at 11. And the rooms are now open.